our today's speaker, Dr. Emmanuel from Illumina, USA. I want to extend my thankful to all the participants and customers across the country in this webinar on the topic Next Generation Sequencing Power for COVID-19 Surveillance. I am compelled to all the participants from different departments, hospitals, institutes, and universities who are joining us today to understand the subject and improve our knowledge about the pandemic situation due to COVID-19. I believe today's session will enlighten all of us and will definitely improve our knowledge and information to combat pandemic situation overall and particularly in Pakistan by using Illumina's technology, next generation sequencing, to have better information of the virus and to take appropriate measures. I would like to pass on to Dr. Ishtiaq Ahmed Khan, who will be acting as a moderator to introduce speakers and also will take note of all the questions from different participants that will be answered particularly at the end of the session by Dr. Zara as well as by Dr. Emmanuel. Thank you very much and pass to Dr.
Bismillahirrahmanirrahim. Assalamu alaikum and very good afternoon. It's indeed a pleasure to host you all, and I am very thankful to Zafar Mahmood Sahab for very kind invitation. Nothing is more well known than the COVID. This is the, you can say that it is post-COVID era, and we are just picking the pieces up and we are trying to understand that what has been done by this pandemic and what is expected. So, as we all know that hallmark of this COVID-19 is rapid human-to-human transmission, and very quickly it swept the globe. So it caught the whole technology scientists and the humanity at large by surprise. I was reading an article in Science, and that was a very comprehensive article about how does the SARS-CoV-2 kills. So we can see that the mode of progression of the disease is quite diverse. It varies patient to patient. It can be from asymptomatic to lethal. And initially it was thought that it is a slow mutating virus and probably we don't need to worry about the rate of mutation. But eventually, like many other expectations, this expectation has been proved wrong, and that virus, due to very high transmission rate, proved to be highly mutating virus. So due to high rate of mutations, we can see that now reinfections are becoming more and more common and new variants are emerging. We are very much familiar with B117 and South African and Brazilian variants. So as per Dr. Gupta of the Oxford University, the vaccine escape is imminent. So if not today, we can expect that probably there are some variants in some other part of the world which are not under the strict surveillance as the surveillance is going on in UK and other advanced countries, but UK is for sure at the top of the list. So if there are unnoticed mutants are spreading in some other part of the world, they can invade and they can catch us in surprise once again. So we cannot overemphasize that the genomic surveillance by using next generation sequencing technology is the need of the hour. It need to be extended in other part of the world as well, as it is going on in UK and other advanced countries. So I'm very hopeful that Dr. Emmanuel Terrison and Dr. Zahra will educate us further about the subject matter, and we will be able to realize the gravity of the situation in a better way, in the need of the genomic surveillance to hunt the new genetic variants and to devise the strategy for better specific diagnosis and eventually the disease management. So without any delay, I would like to invite Dr. Emmanuel Terrison. He is working with Illumina as a application specialist.
Sir, we will
as an application specialist and as well as the
we will first take the presentation of Dr. Zahra Hassan.
Dr. Emmanuel Terrison.
Hello everyone, can you hear me?
Hello, Jess, we can hear you.
Yes, okay, perfect. Sorry, I had some issues with my connection. Okay, let me just share my screen. Okay. It seems I cannot be sharing my screen. Would there be any way for GMS team to allow me to share? Okay, perfect. Can you just confirm that you can all see my screen?
Yes, the screen is visible.
Okay, perfect. So thank you very much for inviting me to be speaking today with you. So the purpose of my presentations today is to show the value of using next generation sequencing in the fight of COVID-19 and specifically around the detection of the new SARS-CoV-2 variants. So my name is Emmanuel Terrison. I'm clinical sales specialist for Illumina. So I will be starting with a brief overview of the pandemic. So as you all know, the SARS-CoV-2 pandemic started so actually late December 2019. And the virus was identified and characterized in January 2020 on Illumina platform. So since January 2020, we have seen an increasing number of cases. So in the case of Europe, around March to April, the number of cases was increasing significantly, and we have seen an emergence of new cases, quite dramatic in Europe, for example, around autumn. So from October to December 2020. And in December 2020, we actually saw the emergence of new SARS-CoV-2 variants. So namely the strain B117, identified in the UK, as well as the B1351 from South Africa. And these new strains have been raising lots of concerns due to the fact that they are highly transmissible. And there are some also worries in regards to vaccination escape, and so on. So today we see an increasing need to be using sequencing to be able to characterize these emerging new variants and to be able to take some decisions around that. So you can see some of the news here from the US regarding the new UK variant. So not that new anymore, actually. So you can see it's been a growing concern since it started in the US, but it is not, of course, not only in the US. So this slide is US centric because Illumina is based, the headquarters is based in the US, but we have seen these new variants emerging all across the world. So you can see on this slide some information regarding, for example, the vaccines that are made available all across the world. And why it is important is because, for example, the so-called South African variant, so B1351, has been actually shown to be able to escape some of the vaccines. So the AstraZeneca vaccine, for example, and this has a major impact at the state level because, of course, in the case of South Africa, for example, the AstraZeneca vaccine has been stopped. The deployment has been stopped in South Africa, and in the country has now changed to the Johnson & Johnson vaccine. So it is important because, of course, emergence of such strain will dictate the decisions that need to be taken at the country level, and it has also a massive impact both on terms of the lives that it can save or impact, as well as on the economy. And you can see here another post, for example, in Europe, where the number of cases emerging with the South African variant are actually also impacting the vaccination strategy. Okay. So the main value of NGS will be to be able to identify these new variants. So if we take a look at the different strains, so the B117 that was originally found in the UK is now present in lots of countries. So at the time this slide was made, it was in 26 countries, but we see it in now much more countries than that. So the main concern with this lineage was actually the number of mutations. So if you can see the progression of the virus along across time, you can see it has an increasing number of mutation, of course, which is natural, but you can see that B117 is presenting a much higher number of mutation compared to what we would have been expecting, and this is the main concern. So originally, one concern was due to the fact that this UK variant has been presenting one mutation, one deletion in the protein S coding gene, and that is causing one of the main qPCR assay, the tag path from Thermo Fisher, to be failing on one of the three amplicons. So this is also a concern because, for example, some of the qPCR assay are actually targeting only two amplicons. So you actually miss 50% of the information if you're using such assay. It also carries mutation in the RBD gene, impacting the binding to the ACE2 receptor. And if we look now at the South African variant, it's been of course detected in South Africa in one of the lab that we are closely working with. It's been of course found in much more countries now. So it has been, after South Africa, it was found in Zambia. It has been moving to the UK, Switzerland, Finland, and much more countries across Europe. One thing to note is that it is not presenting the same deletion as the UK variant. So meaning that using an assay such as the Dropouts assay, you wouldn't be able to differentiate this strain from another. So that's also a major concern, and that is why qPCR cannot be the only strategy that is used at the country level. So on one end, you would be able to let's say to guess that your variant, that the strain might be the UK variant due to this dropout, but you are missing all the strains that are not presenting this mutation. So why are these strains presenting such a high number of mutations? So this is likely due to the fact that they've developed through evolution within a patient with prolonged viral replication. So basically, for example, in South Africa, there are lots of immunocompromised individuals due to the high HIV rate. So meaning in these patients, the virus has been evolving longer, so it had more time to mutate over time. So if we now have a quick look on some surveillance initiative. So you can see here a study that has been conducted in the US. So you can see in this graph that certain samples were tested, and out of these samples tested, you had 0.5% of the samples that were presenting this deletion in the S gene. So meaning that in these 0.5% of samples, there was a suspicion that it might be the UK variant that had entered the US. Okay, so what has been done is doing some sequencing on these samples to know whether or not these samples were actually carrying, these patients were actually carrying the UK strain. So what came out of this is that actually only 32%, only 32% of these samples were carrying the UK variant. So meaning that without an NGS, you would have thought all these 0.5% of the cases would be UK variant, but actually 32% were the UK variant, so the one highlighted in yellow here. So 51 cases, and all the others were other variants. So which doesn't mean that it's not important, but it means that with qPCR only, you would be missing this information. You have no way to understand whether or not it is the UK variant and whether or not it would have a different transmission rate and all the information that you cannot get without sequencing. Okay. So as I mentioned, the South African variant is not carrying this mutation. So with qPCR only, you have no way to know whether the South African variant has actually entered the country. So that is why the sequencing is actually key now to address the pandemic. This study that has been conducted in the US has been published, so it can be found. Some press release can be found on the Illumina website, and there is actually a scientific publication as well. And this study has been conducted using the Covici assay from Illumina, which is a high throughput assay. I will be mentioning a few words on this solution, but it is not the only solution that you can use, and that's what I would be showing you in my slide decks. So another thing that is important is to look at the data that has been uploaded on the GISAID database, which is the main database used to track the pandemic. You can see on these two graphs the data that has been coming uploaded to the GISAID database. On the top graph, you can see data coming from non-Illumina platform, and on the bottom graph, you can see data coming from Illumina platform. So one thing that is important to note is these yellow orange bars. And these orange bars are showing the the number of UK variants that have been sequenced. So you can see that using non-Illumina platform, the UK variant has been detected at week 47, while for Illumina users, it has actually been detected 8 weeks earlier. So that is due to the accuracy of the Illumina sequencing. So you can see that you are able to detect it so faster because of the high sensitivity of our assays. Okay, so now, if we go a bit more into the importance of surveillance and the role of NGS, you will see it's quite straightforward. So the first value of doing surveillance is that it will support allocation of resources, and these resources are multiple. So it's of course it's in terms of human resources. So you need to have people taking care of the patients, and so you have to know where you have to allocate these people. So basically, when you do the surveillance, you can see, you can track the emergence of new clusters, and you can take decisions based on that. So it will help the local and state and government institutions to respond to the emergency. It will, of course, provide information to accelerate developments of therapeutics and diagnostics. So, of course, here we are thinking, of course, of the RNA vaccines, and it will allow the government agency to rapidly implement policies and for prevention. So basically, once you do some surveillance, you can actually take decision at local or national levels. So it can be closing the borders, putting curfew in place, putting lockdown in place at a city level or country level. So really, it's the value of NGS and surveillance is really to provide a full picture, all the information that you need to take informed decisions. So, and why is the surveillance data important? So, of course, at national level, it will allow you to monitor emergence of new important strains, and it will also allow you to monitor the trends after you have taken some measures. So it can be vaccinations initiatives, it can be lockdown, it can be like any measures that are taken at the country level. So you can directly through surveillance study the impact of these decisions and initiatives. At local level, you will be able to identify some new clusters, investigate outbreaks, and improve understanding of local epidemiology. Okay. So now, if we go into the different workflows, you will see there are plenty of workflows, and I will be mentioning specifically 4 workflows that you can use from Illumina. So just a quick snapshot of the different solutions I will be mentioning. So these are the 4 solutions. So one is called the respiratory virus solution. The other one is implicit SARS-CoV-2, the Arctic approach, as well as Covitsik. So you can see that they have been sorted in this table based on their cost, relative cost per sample, as well as throughput. So basically, the top solution will present a higher cost per sample and technically lower throughput per week, while at the bottom of the table, you will be going for a lower cost per sample and higher throughput. So the Kovitsik being the highest throughput solution that you can be using. So so the first one is the respiratory solution. It is a broader solution, and that is why it has a slightly higher cost, also due to the technology it uses. You will see it's targeting 30 viruses, respiratory viruses, and I will be presenting that. It can be sequenced on any platform, but it will have a lower throughput per instrument, which is why it is considered kind of low throughput compared to the other. So you have an indication here on the number of samples per run. The SARS-CoV-2 implicit solution is only targeting the SARS-CoV-2 virus, and it's Also, what I can mention is that all the solutions are providing full genome information for SARS-CoV-2, so meaning that they can all be used for surveillance, okay, and to detect new variants. So the implicit SARS-CoV-2 solution is a full end-to-end solution, just like the respiratory virus panel, meaning that Illumina provides all the reagents that you need to carry out the workflow. Okay. There is another approach that I want to mention. It's the ARTIC approach, which is the most widely used approach at the moment that has been developed by the ARTIC network, and I will be talking about that. It is presenting lower cost per sample compared to the implicit solution due to the fact that it is not a full end-to-end solution, meaning that you have to be ordering on the side primers that have been developed by the ARTIC network and use the Illumina library prep solution with it. So it's not a full end-to-end solution. So so that's why it's giving a better cost per sample. And then we have the Covici Assay, which is an FDA-approved assay in the US for SARS-CoV-2 sequencing. It is a full end-to-end solution with the best price per sample, but it is a high throughput solution. So it is best suitable for NextSeq and NovaSeq platforms. And I will be describing that as well. So if we start first with the respiratory virus solution. So the respiratory virus solution is, as I mentioned, targeting 30 virus families. So you can see several coronaviruses as well as SARS-CoV-2. You have adenoviruses, bocaviruses, parainfluenza, also quite a few influenza A strain here as well, influenza B, rhinoviruses, and so on. So really the purpose of this solution at the moment, of course, is to be investigating SARS-CoV-2 as well as the co-infection. But after the pandemic will have passed, the idea of this solution is to be using it for any patients coming in with flu-like symptoms. So basically, the patient is coming in, you are collecting a swab sample, oropharyngeal swab, for example, and you would be running sequencing to identify which is the virus that is responsible. Okay, so without knowing beforehand which virus it is. Okay, so that's the main purpose, not to have one solution per virus type that you want to test. So, but one solution that would regroup everything. So that's for example a perfect solution if you are working in a lab that is specialized on respiratory viruses. So in the like of the SARS-CoV-2 pandemic, it is also providing high value because it can allow you to be looking into co-infections. So as you can see here, there are some publications showing that we have detected co-infections in 22% of the COVID cases. Okay, so you can see that the most common was rhinoviruses, but you can see that there are also influenza A, parainfluenza, and so on that have been detected. So basically, this is purely on the research side to be really investigating the impact of co-infections in COVID-19 cases. Okay, so that is one solution. It is quite comprehensive, so it's providing really good information. We then have this implicit SARS-CoV-2 solution. So implicit is a workflow that is quite simple to follow. It's amplicon based, so it's PCR based. It is providing full SARS-CoV-2 genome information, but simply SARS-CoV-2. So if you have interest in co-infections, then you would be looking at the respiratory virus solution. If you're simply looking into SARS-CoV-2, then the implicit SARS-CoV-2 is quite a good solution. It does not require too many reads per sample. So as you can see, you can already sequence quite a few samples on the smallest benchtop platform, which is the i600. And it also including, just like the respiratory virus solution, it's also including human controls. So the workflow is quite straightforward. You start from the RNA that you extracted from your swab. You generate cDNA, and then you will do the PCR amplification of the SARS-CoV-2 genome. And then you construct a library, adding the adapters and so on. So the full workflow takes around 7 hours with 1.5 hours hands-on time. So it's quite straightforward. It's a good solution in the sense that it is that you can get all the reagents from GMS in Illumina. So it's quite comfortable solution to use. As you noticed, I mentioned Arctic approach. The Arctic approach is at the moment the most used approach. The reason being that it is an approach that has been basically created by the scientific community, and then and behind that is the Arctic network, which is a network specifically working on infectious diseases and working on designing end-to-end solutions. So basically they work on designing the primers and then working out some workflows that can be used on any platform. So they have been working on Ebola, on influenza, and now on SARS-CoV-2. So they have been highly collaborating with lots of labs across the world, and that is why now it's the most widely used. So basically, with this workflow, what you would be doing is doing a PCR that is local specific of virus of interest. So here, namely, it is SARS-CoV-2. So the primer sequences to be used are provided by the Arctic network. So you just have to go on the Arctic network website and then order your primers from your preferred manufacturer. And then what you would be doing is preparing a library prep from this PCR product. Okay. And then these samples can be sequenced on any of the Illumina platforms. So the main value of this is that it's giving you great flexibility because, of course, at the moment, main focus would be SARS-CoV-2, but what you can consider is doing any type of first PCR step to be targeting any organism that you wish. So it is quite flexible and it is quite low cost because, of course, you just have to buy one library prep from Illumina, and then you have to source the PCR primers and reagents as well as the cDNA conversion kit from your preferred manufacturer. So it's giving you flexibility and good price per sample. So just a few words on this library prep solution to be used from Illumina. It is a library prep that is really used a lot in microbiology, for example, due to its simplicity. It's taking like 2 hours to prepare a library with only with less than an hour hands-on time, and it is using quite a unique technology that is called bead-link transposome. So the bead-link transposome is, you can see a little representation here. It's basically beads which we have enzymes that are bound, and basically that's on these beads that a key reaction will occur that is called fragmentation. Basically fragmentation of your input material as well as adding some tag sequences that are used to create the library with the PCR step. So the value of this solution is that it's simplifying your workflow. So you can easily imagine that when you use a mix that contains these beads, you have a known quantity of beads in this mix. So that means that once you reach a point that we call saturation point, meaning that all the beads have captured all the DNA they could, you are in a situation where your library will be the quantification and normalization of your library will be optional. So if you have been using NGS, you will know that one critical step is to quantify your library once you have prepared it. and then to normalize it, meaning that you would dilute all your libraries to have similar concentration for each libraries to be able to pull them together in a sequencing run. So all of this is done for you with this solution, because you will always get the same quantity of libraries at the end of the reaction once you have reached this saturation point. So you don't need to quantify and normalize. You just have to pull all your samples by volume. So it's simplifying your workflow quite a lot. Okay. So this solution, for example, has been critical in South Africa in order to characterize the South African variant. So we have been working quite closely with a lab that is called CRISP in South Africa that is using exactly this protocol and the Illumina library prep to do this workflow. So they started using this Arctic protocol with the Illumina DNA prep sometime, I think it was around May or something like this last year. And they now have been processing more than 2,000 samples on their MySeq. So, and this is so why I'm mentioning it is because that's the protocol they actually used to characterize the South African variant. Also, what is quite good is that they have published their protocol on a website that is called protocols.io that you might know of. So it's really easy. You can just go on protocols.io. You have the links here, and you can just go there and follow exactly their protocol to be characterizing SARS-CoV-2. Okay. So the last solution I wanted to mention is the Illumina Kovitsik assay. So it is a critical solution now to be taking on genomic surveillance initiatives at national level. So the scale, of course, is different, and that's what makes the test affordable. It's because it's really high throughput. It has been FDA approved with the emergency use authorization for diagnostic, of course, applicable only in the US. So it is a great tool for surveillance at large scale. It is providing, of course, full SARS-CoV-2 genome information. It contains human controls as well. So if you remember what I mentioned about the Arctic approach, it's basically similar to the Arctic approach as just mentioned, except that the solution is a full end-to-end solution. So we are providing all the reagents, so from the cDNA conversion kit, all the PCR and Arctic primers. We are also providing some positive RNA controls as well in the kit. So it's really full end-to-end solution that is really aiming at doing large scale studies. And when I mean large scales, it's at least 384 samples on the NextSeq platforms, and up to 3,072 samples on platform like the NovaSeq. So of course, you can be running this almost daily, so you can be getting up to 3,072 samples per 24 to 36 hours. So it's really high throughput, and that's what makes it also quite affordable in terms of cost per sample. So if you look at the workflow, it's you can see all these 6 steps. So the first step would be, of course, as any other workflow, RNA extraction. Then you are converting your RNA into cDNA that you split in 2 pools to do 2 PCRs, and then you are mixing together your PCR products. So the PCR product, you can see the information on the 2 pools here. So it's similar to Arctic approach. It's a tiling approach all across the genome of the virus. So you have 2 pools to be able to do that in the best way. So once you have done this PCR, then it's a similar library prep approach as I described earlier with the bead-link transposome to fragment and tag the PCR product and add the adapted sequences. then you do a PCR, and then you would be pooling your libraries together by volume. So you pool the libraries by 384 samples to be running on the NextSeq 550, and then you would be doing 8 of these pools to be sequencing up to 3,072 samples on the NovaSeq. Okay, so once you have pulled your libraries together, you would be quantifying, diluting the library pools, load onto the sequencers, and then run the data analysis. And the data analysis is what I will be describing quickly now, because it's also quite critical. And we have developed some tools for that we are actually providing free of charge to any of our users. So the solution that we have developed is called the Dragon Lineage app. That is available on the cloud, free of charge. So what this app will do is it will generate a consensus virus sequence, so a FASTA file. It will generate a lineage data and clade data that are from these online tools that you can see here, the Pangolin tool that has been developed by the COG UK consortium, as well as the NextClade tool from the NextRain database. So basically what you get out of this analysis is the information everyone is looking for, so the lineage information, and then you have some phylogeny information to be looking at where are clusters that have been detected, phylogenetic tree, and then information on the mutations that the strain is carrying. Also, we have made other tools available that are allowing you to directly upload and streamline your upload to the known databases, which are Gisaid as well as NCBI. So just a quick representation of these tools. So an assignment of clades and mutation using NextClade. So it is a website that you can use also outside of our cloud. So basically you just upload your FASTA file and it will be showing you all the mutations that have been detected. So whether or not there are substitutions, deletions, and so on. And the other critical information is from the Pangolin tool that is basically adding a lineage to your sample, and then you have some nice maps that you can use to be seeing where are these lineage detected, in how many cases, in which countries, and so on. So it's quite useful information, and it is also available as a free tool online. So just a few conclusions word for my talk. So basically, genomic surveillance is providing key information to identify and track emerging pathogens. So of course, at the moment, it is SARS-CoV-2, but it is not strictly limited to SARS-CoV-2. Full genome information of any pathogen can be used in this fashion to be conducting genomic surveillance, and it is quite critical. So the Illumina protocols that I've been mentioning, so the 4 protocols I've been mentioning, all provide you with full SARS-CoV-2 genome information, which are providing key insight on epidemiology of new variants, including the new variants that everyone cares about, the B117 from UK, the B1351 from South Africa. The Illumina free app, lineage app that I've been showing, is providing you with consensus sequence, so FASTA file that you can be using to upload on any database. And we have included in this app some outcome from some of these online tools, which are Pangolin as well as NextClade. And of course, we have been simplifying the upload to databases such as Gisaid and NCBI with our tools. So that was all I had to present today. So thank you really much for your attention, and do not hesitate to ask any question.
Thank you very much, Dr. Emmanuel Terson, for your very good presentation. So there is a quick question from Dr. Shakil. He wants to ask you that given the high mutation rate in SARS-CoV-2 genome, what if there are mutations in the sites where amplification primers are needed? So this is a very general, yeah.
That is a really good question, and it is actually, yes, it is a key question. So of course, as you know, if there is mutation underlying some primer sites, we might see some drop of some of the amplicons. So at the moment, it is closely monitored by the Arctic network, as well, of course, as the Illumina team when it comes to Covici assay. So at the moment, what we see is that there are no dropouts. So although there are quite a high number of mutations in some of the strains, these Arctic primers are still able to capture the full genome information. But of course, over time, we will see some drops of amplicon, and this is monitored. And of course, solutions will be adapted to be still capturing the full genome information. Also, what I did not specify is that, for example, the respiratory pathogen solution is not PCR based, it is enrichment based. So meaning that we are generating a full genome library, and then with probes, we are fishing out the SARS-CoV-2 sequences. So only with several probes. So it's not PCR based, so it will be more robust in regards to mutations. So it will be more stable over time.
Thank you very much.
May I intervene a little bit, because there, as per plan, Dr. Ishtiaq, first of all, we will have a session and we will have a speech by Dr. Zara Hassan, her presentation, and question answer will be at the end. So you can write down all the questions, and then we can put the questions to Dr. Emmanuel at the closing of the session, before closing of the session. So first, now I will invite Dr. Zara Hassan for her presentation. Thank you.
Assalamu alaikum and good afternoon. Thank you for the introduction. And I'm grateful to Mr. Zafar Mahmood, GMS, and Mr. Fahad Kamar, who's always been a facilitator for Illumina products, and Dr. Ishtiaq Ahmed for introducing this talk. So I will start what is now a backstory to explain the Pakistan situation, because we have had a fantastic talk on where we are with regard to Illumina products and the importance of genomic surveillance. So I hope that by taking you through our own limited experience, we will see how it's important to be studying variants in Pakistan as well. But so I'll start with, I have no conflict of interest to declare. And so my perspective comes from, from somebody who works not just in research, but also who works in the diagnostic laboratory, and we've been very involved with SARS-CoV-2 diagnostics. So over a period of timeline, as we know, the coronaviruses have had multiple epidemics over the last 10 years, but nothing has been as potent as the current SARS-CoV-2 epidemic. And we know that the viruses are related to human viruses, but the SARS-CoV-2 is sort of different because it was identified in Wuhan in December, as early as December 2019. And then the sequence, which became available, was what has powered the diagnostics and this information that we have had about the global spread of the virus. It's essentially a respiratory virus, which is why it has had such a big impact. And it's nothing compared to the previous pandemics we've had, which, compared with what we had before with regard to SARS-CoV originally from 2005, which only maybe had 7,000 to 9,000 deaths. So we already are at, 114 million cases worldwide and 2.5 million deaths. And this is a global statistic that gets updated every day. And Pakistan is one of those countries that have been watched very carefully because it's a very populous country with a high infectious disease burden. And there have been, variable measures taken with regard to identifying the spread of COVID-19 and trying to manage it. So what we know so far is that we've had 2 waves, one as early as March that went on until June, And another one that we have now seen as a resolved second wave, and let's hope we don't have a third wave. We know that about 581,000 cases have been identified with 12,000 deaths. So overall, we've done quite well in the context of the very severe morbidity and mortality that has been seen globally. I'm speaking from Karachi, which, as you know, is the most populous city in Pakistan, and Sindh has had the largest number of cases, and Karachi has the greatest concentration of cases. And the Al Khan University was one of the first to set up diagnostic for SARS-CoV, and I know that Dr. Ishtiaq's lab at Karachi University has also been involved in diagnostics. And so I want to talk a little bit about, when we talk about, surveillance, who do we need to survey? We need to think in terms of epidemiological context of the disease. What is the age group of people we need to be looking at? Who is transmitting the disease more? Who is being affected more about the disease? Because these are things we need to think of in a lower resource setting, where if we go about trying to identify spread of virus even, or curating it, we'll have to have a plan as to understand who are the individuals that we should be testing and tracing. So most of the individuals that have in Pakistan are found to be male. Most of the COVID is diagnosed in males, probably because of more testing done in males, even though the rates of disease are equivalent, but there are more cases reported in males. Like in most parts of the world, the morbidity is much higher in those aged 50 and up. So what do you need the diagnostics for? We need the diagnostics for both diagnosis of cases and clearance. And the trouble with transmission of SARS-CoV-2 has been that it occurs in both asymptomatics and pre-symptomatic and symptomatic individuals. So the classic, identification of symptoms based on a clinical history is very difficult, because not everyone, because the symptoms are common with those of influenza, so they don't stand out necessarily. And then secondly, because we know that asymptomatic individuals transmit very successfully. In terms of understanding the virus itself, there's been a rapid information exchange globally, with a lot of science about, where we've learned about the biology of the virus. Why is it so transmissible? We know that, for example, it has a certain structure, and the spike glycoprotein is very important to the way that it enters the cell. And then once it's inside the cell, it can reprogram the host, so it can generate and replicate, and then basically come out of the cell and infect other individuals, other cells, and be spread. So in terms of diagnostic timelines, I put this up again because this sort of always needs to be kept into context when we're talking about testing, because there's a pre-symptomatic stage for several days when the virus is present, and then there's a symptomatic and post-symptomatic stage where the virus is also present. So what we did at AKU was we spent a lot of time optimizing testing, and because you need appropriate samples, and even downstream, if you want to work with virus samples, you need high viral load, good respiratory specimens, and the specimens need to be appropriate to the site of disease. So in most cases in early infection, the upper respiratory specimen, which is the nasopharyngeal or nasal or the oropharyngeal specimen, is ideal. But in individuals who have had progressive disease, it is the lower respiratory specimen, which may be a BAL or a sputum specimen, that is better. We also made our own swabs in earlier on in the pandemic, when there was a need to import supplies and there was shortage of supplies. So we made our own locally manufactured swabs using a 3D printer, and these worked quite well as well. And another thing that we did, which has been very helpful for us to scale up testing, is that we have started self-collection of nasal swabs, so nostril swabs or anti-nair swabs, which makes it safer than nasopharyngeal because they're less invasive and they generate less aerosol. And we've also shown that saliva testing works as well. So that's a little bit about background as to how to get appropriate samples. But as introduced by the last speaker very nicely, there's a lot of information about the changes in the virus and the need to try and understand this. Now, the changes in the virus result in mutations. We know that there are several open reading frames encoding for both non-coding genes and non-structural and structural proteins. So of the structural proteins, there's the S, the E, and the M. And then in the non-structural proteins, it is the ORF1A and the ORF1B that have most of the mutations. So now we have, in our limited experience, sort of almost muddled around trying to play with different Illumina protocols, because I would really love to try out the new Illumina protocols that were described. We haven't purchased them yet, but I really do want to use them, because what we have had is that we've used, RNA converted to cDNA, and we've used the Nextera prep. And I'll share some data from our early work, which is we're in the process of expanding, and where we have got some initial data from of SARS-CoV strains from Pakistan from the beginning of the pandemic. So just to put out there that one thing we learned is that it's very important to have good quality RNA, because unless you have high quality RNA, usually from a sample that has a low CT, less than 30 or so, it's difficult to get a good library prep for sequencing from that. The Illumina platforms have been explained very nicely already. I'll just put out there that the MinI and Oxford Nanopore platform is another platform that we have some experience with, and we're, again, trying to optimize our own workflows. So I'll talk a little bit about some work we did, and I'm acknowledging all my colleagues in the laboratory, Dr. Asghar Nazid, Dr. Najia, and multiple colleagues have been involved in both the genomics and both the sequence analysis of the strains. So one of the things that we learned was that the virus evolved. It evolved, and then it became a little bit difficult to follow the evolution, because there were many lineages. So the original lineage that was identified in Wuhan was, didn't have any mutations, but then as mutations developed, first GISAID and the next strain developed naming platforms for the different clades. So we know now that, for example, there's the S clade, the L, the V, G, and then the G, which is now also called the B1 clade, according to the pangolin lineage, is the one that has emerged as the predominant lineage worldwide. So it's sort of a moving scale where the strain nomenclature is changing. We started off with a particular understanding of the strain and a belief that the primers that worked initially would continue to work, the tests that work would be the same. And now, as we're learning more and more a year into the pandemic, we're now worrying about mutations. And it's important to understand these and to track them, because it is the changes that have helped classification into multiple lineages. So this is a simplistic way of looking at it, that the lineage S split to L, the L went to V and G, the G clade went to GH and GR, and so on. So as I said, we used a RNA converted to cDNA and an Nextera platform, and then we used the Illumina MiniSeq and the MySeq platforms. So just some initial data from our strains. We studied samples that were from travelers who'd come to Pakistan in March. March is April, so there were Pakistanis who had traveled to Iran and Iraq and Saudi Arabia, and we immediately saw that the phylogenetic analysis showed that they were quite spread out. We found that the strains are similar to strains found in Saudi Arabia, some that are found in the US and in India. And it's sort of typical of what we understand of the diversity of the virus. We've done some initial next strain analysis as well, pretty much showing the same thing, that there are ways of looking at phylogenetical relatedness between samples, and they match samples across different parts of the globe. The information that we have is again growing. So this is just really a starter, because if we have a small data set that we are in the process of understanding to try and understand the numbers of variants in each coding region of the virus, to try and understand how many are synonymous and how many are missense variants. The synonymous variants, as you know, don't necessarily have a functional effect, but it is the missense variants or the deletion variants that have been known to play a role, because the moment, particularly if they are in the S gene, as we have seen, because the S is the spike glycoprotein, and that is the protein very important in the virus interaction with the host cell. So the most predominant clade, which is the G clade, which is typified by the D614G mutation, was first identified as a mutation that nobody thought too much of. It was identified in Europe and was found to be highly transmissible. But at that time, the scientists didn't even want to think about it being clinically relevant. In fact, the in vitro studies were thought to be not robust. And now we know that in fact they were true, because globally it is the 614G mutation that has evolved into multiple lineages. So in Pakistan as well, we saw that even earlier on, we found the GH clade, we found the S and the L clade, and now what we are seeing is more of the GH clade. So we see, even in the limited analysis we have done, and we've probably looked at 40 or so samples, so very small data set, which we're expanding now, but it identifies that we are definitely seeing more GH clade samples. We also have, through another set of analysis, identified UK variants, so the B117 lineages in travelers, and now, how those strains transmit, how they spread, that's a space that really needs to be watched. I put this up there because we have already heard a lot about the new variants, the new mutations, but to put it into context, I just want to point out something regarding this mutation where D614 becomes fixed, because this is the G clade or the GH clade that then continued to acquire additional variants. And it is these B.1 lineage strains that are most common globally, now most common in Pakistan. They've been shown by other laboratories as well to be spreading through the country. And we don't know how the variants will be managed. We don't know how they are in the population right now. We don't know where they are in terms of numbers, and it is important that through sequencing efforts, we try and understand transmission and also the presence of variants that potentially could cause more morbidity and mortality in our population. So I'll end by saying that certainly rapid large-scale virus sequencing is very important to try and understand the dynamics of virus evolution, and it will be very helpful to try and understand how to manage and the COVID-19 pandemic. And we are at a stage right now where things are opening up. This week, all schools opened up, and by the end of March, everything will be mostly completely open. So probably sooner rather than later, we need to try and get a sense of a snapshot of the variable SARS-CoV transmission variants in our population, so that moving forward, we'll have a better handle. on how to manage the pandemic per se. So our work received funding from health security partners and the AKU, and would not have been possible without support from the Khan University Department of Pathology and Laboratory Medicine. I end my talk here. Thank you very much for listening.
So with that, we will go ahead and take some time for questions. One question is for Emmanuel. Given the high mutation rate in SARS-CoV-2 genome, what if there are mutations in the site where amplification primers are needed? Emmanuel, this is for you.
Yeah, so I actually already addressed this question. So yeah, as I mentioned, this is bound to happen. But at the moment, so when it will occur, we will be losing some of the amplicons where the mutation is underlying. But this is closely monitored, and the primers would of course be adjusted at that time. It is closely monitored by the Arctic network, for example, and I see that there is also the similar question in the chat. So how routinely primers are calibrated to overcome dropouts. So this, how routinely, I don't really know. So for example, if we take into account the primers developed by the Arctic network, we are currently at the V3 version of the primers, but it has been, the V3 primers have been used for I would say, yeah, way more than 6 months now, and they are still working fine. So yeah, I wouldn't be able to comment how soon it will be updated and how often, but at the moment the current version is working perfectly fine.
Okay, over to Dr. Ishtiaq. Okay, so there is a general question for both of our speakers, and that is how routinely primers are calibrated to overcome real-time PCR dropouts, since SARS-CoV-2 houses rapidly changing genome, and what impact does it create for diagnostics?
I can take that question. It's a very good question, because this is something we weren't worried about before. But so when the diagnostic platforms were okayed by CDC and FDA cleared, they all required that at least 2 genes be involved. There should be 2 gene targets, and I think that there will be a need to modify the diagnostics so additional genes are present. So for example, the TACPath COVID assay by Thermo, which has been used as a screening assay, has 3 targets, and it is quite a robust assay. So I think that moving forward, there will be more emphasis on diagnostic assays that have more than 2 targets, because currently assays are recommended to have 2, but I think they will need to have more than 2 because of this reason, and they will have to be modified.
Okay, so there is one question for Dr. Emmanuel, and that is how Illumina is unraveling the host immune response using the kits, the solutions they're providing.
So sorry, I missed a bit part of the question. What was it again?
what solutions Illumina is providing to unravel the host immune responses.
Okay, so in regards to host response, so there are two different type of approach that can be conducted. So one would be to be looking at the RNA, so the transcriptome of the host. For example, you can be conducting a full RNA-seq of the sample, which would be giving you both the virus genome as well as the host response to it. So there have been quite a few studies conducted on that. For example, quite a few conducted in the Middle East, for example. And that's basically standard RNA-seq that would be capturing both the host and the virus. The only concern is that you need quite a lot of reads per sample to be getting a good picture of the host response. There are others initiatives that are conducted, so not on the host response, but really on the host genomic makeup with for example genotyping approaches. So there is a consortium working on that in Europe, for example. So these are the two type of approach that are conducted on the host.
Okay, there's one another question. Are there any specific regions in COVID-19 genome which plays a vital role in disease transmission and contagiousness?
Yes, it is the spike protein, which is responsible for binding to receptors on the host cell. And that seems to be key because it was the modification in the spike protein at the 614 codon, as I mentioned, the G to D change that resulted in a higher viral transmission and more shedding. So the spike protein, we know, plays a role in that.
So I think we don't have more questions. Maybe if we have questions, we will email to Dr. Emmanuel and maybe Dr. Zara by email, and then those questions can be addressed directly to the customers or to the other sites. So at the end, thank you very much, all the participants, especially my gratitude to Dr. Zara Hassan for her time, and Dr. Emmanuel from Illumina, Dr. Ishtiaq from Covici University, being with us today for the session. It has been very much enlightening session for all of us. And from the GMS platform, I would like to say thank you, and will like to say my thankful, as well as I commit to continue such activities with your collaboration and with your cooperation. Thank you very much.
Thank you.
Thank you, everyone.